And if all goes well, it should be visible. Yeah, I think it is visible. Perfect. All right. So I'll be using some time to share a bit about the latest R&D work we've been doing at Nalagenetics. But very briefly, just to introduce ourselves, we're a biotech startup based in Singapore. And we focus on developing genetic testing solutions for preventive healthcare. And this includes the area that I'll be focusing on today, which is pharmacogenomics. So what I've done for my talk is I've structured it in two sections. I'll start with a bit of background, just introducing the idea of pharmacogenomics, highlighting some of the solutions that we've been developing to promote its adoption, and also mention some of the programs that we've been able to participate in that are driving PGX implementation in Southeast Asia. And then I'll jump into discussing our latest R&D work on the long-read space since we became ONT users last year. And this is going to be the majority of my talk. As it's been mentioned, if there's any questions, please drop them in the chat and we'll be addressing them at the end of the webinar. Okay, so without further ado, I thought I would start just opening with a couple of stories that I think illustrate quite well the use case of the test we'll be talking about. So here on the left, we have the story of Mr. Tan. He had been diagnosed with chronic gout, and as such, he had been prescribed a common medication to lower uric acid levels called allopurinol. However, unfortunately, just briefly after starting to take that medication, he started to experience some unusual symptoms: skin rashes, blisters, fever, fatigue. And so he had to go back to the emergency room, where eventually, after some testing, he was diagnosed with Stevens-Johnson syndrome, which is a life-threatening reaction to allopurinol. He could be treated, but this diagnosis actually left him with some long-term complications. Now, here we've got another story by Mrs. Lim. She actually has a history of cardiovascular disease, and after some complications, she received a stent. And to prevent any blood clotting forming around the stent, she was prescribed clopidogrel, a common blood thinner. However, even though she kept sticking to that treatment and she kept taking the medication, she had to rush back to the hospital 3 months later with symptoms of another heart attack. And at that point, she was diagnosed with a blockage around her stent, suggesting that the treatment with clopidogrel actually had failed to achieve the intended goal. So what do these two stories have in common? Actually, both of them talk about suboptimal responses to treatment. Both of them have an underlying genetic cause. And most importantly, both of them could have been prevented by having access to a genetic test to check for these underlying mutations. So this is exactly where pharmacogenomics comes in. Pharmacogenomics, very briefly, just looks at how genetic variants can impact the way we respond to drugs. And it's often highlighted as one of the key or poster examples of personalized medicine because it has this capacity to bring us from a one treatment fits all approach, which is common practice nowadays, to a more personalized approach where each patient would be given customized treatment depending on the predicted response to drugs. Now, the other interesting thing about pharmacogenomics is that there's some key characteristics of pharmacogenomics variants that make it a very good candidate for adoption at scale. And these are the following ones. So pharmacogenomic variants are highly penetrant. We've seen a couple of examples on how they can be linked to adverse drug reactions in the previous slide. But not only that, they're also highly prevalent in the population. In fact, most of us will have at least one variant in one of the key pharmacogenomics genes. And then lastly, they're also actionable. Nowadays, there's already clinical guidelines that dictate the next steps in case we have a specific pharmacogenomic variation. And this could be either changing prescription or adjusting the dose. And there's even studies that have shown benefits to applying this type of genetic testing and impact in terms of reducing adverse drug reactions. So this is where NALA comes in. One of the areas that we work in is in developing solutions to help our partners to adopt pharmacogenomics. And this is the case, for example, of NALA RxMax. This is the solution I'll be focusing on today. It's our most comprehensive PGX report to date. In a nutshell, it covers 35 pharmacogenes. including over 350 gene-drug interactions. And it's also platform agnostic. It can be run from a wide range of inputs from different genetic testing platforms. However, today I'll be focusing more on the long-read aspect of it. Now you can see here on the right an example RxMax report. This is actually just one of the pages for a specific statin. And we can see how in this case, the patient was recommended to take an alternative drug because they were found to have decreased function for the metabolizer enzyme. So this is the type of insights that you can get from this type of tests. Now, we've been lucky enough to be able to work with different partners, over 20 different partners in Southeast Asia mostly. And this includes population sequencing programs in both Indonesia and Singapore. So I just wanted to share a bit more about what these programs have consisted of. So in the case of PGSI, the PopSeq program in Indonesia, they are currently using our RxMax algorithms to generate customized reports for 10,000 of their study participants. And you can see a bit of a snapshot of the customization with it over here in the left. And then we also have another use case from Precise, the population sequencing program in Singapore, where in that the challenge was a bit different. They already had a system set up to do the reporting. However, because the public healthcare system in Singapore is fragmented, there's 3 main healthcare clusters, their need was mostly around the return of reports to study participants. And doing so in a way that was safe, but also in a way that enabled the participant to then share the report subsequently with other physicians, be it from public or private hospitals. So that's the work we did with them. We developed a mobile app to enable this type of return of results. Now, another one that I also wanted to mention is a more recent implementation study that we've done in partnership with Raffles Medical. So this is a private hospital in Singapore, and this study has just been published as of a few days ago. So you can refer to the paper here for further details. But essentially, this allowed us to learn some additional numbers or some additional figures on the clinical utility of adopting PGX more in the local setting in Singapore. So just to share some of the findings with you today, some of the things we learned was that, as expected, the vast majority of participants had at least one actionable pharmacogenomics variant, so up to 95%, and this was very much aligned with what we had seen in other publications. Now, notably, this information was immediately useful for the participants. Over 20% received actionable recommendations for the drugs they had been prescribed. And then perhaps most importantly, in the 12 months follow-up survey, we saw that 17% of the remaining participants had actually made changes to their medication following receiving the PGX test. Now, unfortunately, we didn't have the chance to look at adverse drug reactions in this study or see a decrease around those, but still it provides quite useful information around the actionability of PGX testing, again, in the local setting in Singapore. Okay, so I've talked around the different use cases of pharmacogenomics, different implementation studies that are happening in the region, and how we can go about sharing back this data. But what are the challenges that lie ahead? Now, from more of the technical perspective, one of the challenges around PGX testing is that identifying variants in pharmacogenes is not always an easy task. And this is because of different reasons. So number one, these pharmacogenes, they tend to be highly polymorphic. So if we look over here, this is just a panel illustrating the variation that we see for CYP2D6. It's one of the key pharmacogenes. It's responsible for metabolizing more than 20% of drug in the market. And we can see how on top of typical SNP and indel variation that we would typically see, it's also prone to having a lot of copy number changes, hybrid rearrangements with downstream genes that are very similar in the sequence, and then even a combination of both, so both hybrids and copy number changes. And you can imagine how this makes it very challenging to resolve the actual underlying variants just because of this complexity. The second reason why it's not easy to genotype pharmacogenes is because it's not just about finding the individual variants, but it's about phasing them into haplotypes. because eventually it's those haplotypes, the information that we use to derive a function for that specific gene. So just to illustrate with an example, we've got here CYP2B6, and we've got two SNPs, but we can see that depending on the haplotype that these SNPs fall in, be it in the same or different haplotypes, they actually can be interpreted as different star alleles. And not only that, that interpretation, star allele assignment, will also impact the phenotypic consequence. So therefore, it's not just important to learn which are the variants present, but we also need to understand how they're phased, how they can be resolved into haplotypes. And then lastly, and probably also overlapping with the other two, we know that pharmacogenes tend to fall within complex genomic regions and tend to have rare and novel alleles. And just to illustrate this point, I've included this table from a publication. We can look, for example, at CYP2D6, the same gene that we highlighted in the first panel, and we can see how 100% the full locus actually overlaps with what's considered a complex region, and that over 97% of known variants for this gene actually are classified as rare variants. And what's most notable is that this is not actually an exception. It's not just this gene. This is quite consistent across all the other pharmacogenes highlighted in these papers. So that's actually what brought us in our clinical lab to, about a year ago, consider using ONT, long read sequencing, to see if we could actually improve the accuracy of our pharmacogenomics tests. And specifically, we started looking at a method from ONT called adaptive sampling, which is a targeted approach to doing sequencing. So very briefly, how this works is adaptive sampling actually takes advantage of the real-time sequencing that happens with nanopore sequencing. So as DNA molecules cross the pore, we're able to read the DNA sequence, and then the sequencer is also able to do alignment against the human genome in real time. And so with this information, what we can do is we can provide the sequencer with a list of targets that we're interested in. And so in real time, the algorithm is able to understand whether the sequence that's crossing the pore is actually aligning to our target candidates, to our regions of interest, or not. So if it matches our regions of interest, then it will continue the sequencing. The fragment will continue to be allowed to go through the pore. But if it does not, at that point, then the DNA fragment can be ejected from the pore, allowing a new DNA fragment to land in. And you can continue doing that for a period of time. So what this results in is a data profile like the one you've got at the bottom, where we have an enrichment of long fragments in the regions of interest. We call these on-target regions, but also because we're having to read a small stretch of DNA to make that decision on whether to keep or eject the DNA fragment, we also end up with this genome-wide off-target signal, which can also be used for downstream data analysis, as I'll show in a few slides. So the nice thing about this method is that it doesn't, even though it's targeted, it doesn't require any customized library preparation. All of the selection happens during sequencing, so it's very flexible. And the reason why we looked at it as well, instead of doing whole genome sequencing, is because it also allows us to be a bit more cost efficient through multiplexing. Because it's a targeted assay, we can start having different samples, different libraries together in the same flow cell. At the moment, we have been running this in a 3-plex format, but actually there's room to grow that and therefore reduce costs. So this is some of the ongoing work that we have right now. So having introduced the method, how our workflow looks like is we start from blood samples. We have a few hours of sample preparation and library preparation, followed by 3 days of sequencing and a couple of days of data analysis. And then at the end, we obtain a clinical report like the one I was showing before that will cover the diplotype, phenotypic interpretation, as well as recommendations from guidelines for 35 target pharmacogenes. So we're targeting more regions with this assay, but actually in terms of PGX reporting, we're interested in 35 genes. And this would cover the tier 1, very important pharmacogenes from PharmGKB, as well as HLA-A, which is a gene that we were already testing in our lab. So that's about the workflow setup. Now, something else we did was we ran a set of validation experiments to be able to run this workflow in our clinical lab. In a nutshell, we did a total of 10 runs and covering a total of 17 unique samples, which includes both cell lines as well as clinical samples. And this is going to be the results that I'll be focusing on for the rest of the talk. But just to give you a gist of the type of studies that we've been looking into. So number one, we've been looking at limit of detection, where we were evaluating different DNA input amounts, as well as different base calling modes. For those of you familiar with ONT sequencing, we looked at the high accuracy and the sub-accuracy mode. And we were evaluating the impact of each of these settings to variant calling performance, both at looking at small variants as well as large variants. So that was the first study. Next, we also looked at accuracy assessment. And we did that using a combination of cell lines and clinical samples. And in this case, we were taking things analysis all the way to the end. So we were looking not just at individual variants, but diplotypes and phenotypes. And we were evaluating the concordance against the truth set and against orthogonal methods as well, where the data was available. We also looked at precision, comprising both reproducibility across different replicates and repeatability. So what happens when you run data analysis multiple times using the same inputs. And lastly, we looked at specificity. So over here, we looked at the impact of spiking in some interfering substances, as well as the presence or absence of cross-contamination. So what I'll do next in the next few slides is I'll expand a bit more into each one of these experiments. Okay, but first, I also wanted to share some more general stats about the type of throughput and type of data that we were getting from the adaptive sampling workflow. So what we can see here is that on average, across the 10 runs that we did, and each run was run in a 3-plex fashion, on average, we were able to get an on-target coverage of 25x, off-target coverage of 3x, and this reflects an enrichment factor of around 8.5x. So that is telling us already that adaptive sampling is working as intended. We get higher coverage on the on-target regions compared to the off-target regions. But we can also see the same trend here when we look at N50s. So when we look at the on-target regions, we can see how we're getting long reads. In this case, the median length is around 8 kb. Compared to the off-target regions, we're getting around 640 base pairs only, so much shorter to the range of short read data. So all of this was very reassuring to see. It's telling us that adaptive sampling is working as intended. So with that knowledge, we went ahead and started looking at the results of each study. For limit of detection, here we were focusing on first looking at the variant calling performance, but looking first at SNPs and indels, small variants. And we were doing that by looking at our target pharmacogenes. So here you can see the results in the table below for the different metrics that we were evaluating. And as I mentioned, we were evaluating also a couple of settings. We were looking at different DNA inputs and different base calling models. And so in a nutshell, what we could see was, number one, actually not much difference between DNA inputs. Results remained fairly consistent, but we did see a slight improvement in performance when adopting sub-calling compared to hack. This was most pronounced for callability over here. So on the basis of that, sub became the candidate model to adopt by default. And actually, when we looked at large variants, structural variants, that allowed us to confirm it. So for structural variants, what we did is we looked at different event types, namely deletions, duplications, and hybrid events. For two specific genes, 2D6 and 2A6, because these were the genes for which we had samples with this type of events in our data set. And then we looked at the same two settings, different DNA inputs, different base calling models. And again, very similar to what we had seen with short variants, no difference between DNA inputs in terms of performance metrics, but a bit of a difference in terms of the base calling method. In fact, we were able to see one false negative deletion that was present or was labeled as false negative with hack, but we were able to rescue that with SAP. I'll also note in this table that down here, we've got still 2 false negatives for hybrid events, consistent between HAC and SAP. But upon looking at those a bit more in detail, we actually saw that these were a bit more linked to differences in sequencing depth. So the sequencing depth for these samples was slightly lower because we couldn't see those for the other DNA input, lower DNA input. So from that, actually, we concluded that for SV detection, sequencing depth, it's a bit more sensitive to sequencing depth. So it's important to reach high on-target depth for that. Okay, and then the last thing I'll mention for this slide is that notably there were no false positives detected, and this was very reassuring since we were thinking of implementing this workflow in a clinical setting. Right, so these were the results of the LOB study. They allowed us to eventually focus on the SAP-based calling mode and narrow things down to that and show that the variant calling performance was at a high level, both for small variants and large variants. So what we did next was to extend the analysis all the way to diplotype calls. And we did that in three sets of samples. On the one hand, we had reference cell lines with TruthSet data. We had also clinical samples from one of our collaborating hospitals here in Singapore. And then we also reprocessed a subset of our EQA, external quality assessment samples. So what you can see here is for the different genes evaluated, we can see the diplotype calling results across each of the samples, quite small. Sometimes there's going to be multiple replicates, but in a nutshell, what we can see is that from this evaluation, the majority of results actually came out to be concordant with the TruthSet. There was a certain number that actually were initially discordant, but after reviewing the results in more details, they were reclassified as improved. You can see it's a significant portion there, and I'll show some examples in the next slide of what these improved calls look like. And then there were also some that remained as no calls, so we weren't able to resolve a diplotype. Those, again, were mostly attributed to lower depth. Lastly, we also found 2 cases of discordant calls, which we attributed to poor variant calling, and also I will also share these examples in a few more details in a couple slides. But in a nutshell, you can see how the majority of the calls were either concordant or improved, actually totaling to about 98% of the calls. And we could actually see improvements in 11 out of the 20 evaluated genes. So quite good results so far. So just delving a bit more into the improved calls to give you a glimpse of what they look like, they mostly fell into 3 categories: either better resolution when it comes to phasing variants. We came across a few cases where we could identify novel alleles, and then some other cases were related to expanded haplotype coverage. And what we mean by that is, because this is a sequencing-based method, it's unbiased, right? So we were able to see all of the SNPs or variants in a given region, compared to sometimes the TruthSet was established with methods that like microarrays where there had been a pre-selection. So it's just that we were able to detect variants in an unbiased way. So a couple of examples. We've got here NAT2. For this gene, actually for this specific sample, the TruthSet GetRM was giving an ambiguous call. It was the same for actually short read data when analyzed with DRAGEN and PyPGX. There was no full resolution of the diplotype. Stellar PGX with short read data was actually returning incorrect star alleles. But actually with long read sequencing with adaptive sampling, we were able to resolve the star alleles down to suballele. But most notably, we obtained very clean phasing information. And hopefully you can see that from the plot where we've got in the first haplotype, this is star 5002. We've got these 2 variants in the middle. So we've got this variant here and this variant here, nicely phased with the long read data. And then for haplotype 2, star 6002, we've got these 3 variants which again, you can see nicely phased through the long read data. So this is one example where we saw much more clear phasing. And then the other example I also wanted to share is CYP2B6 for this specific sample here, where the truth set data, microarray, short read sequencing data were fairly consistent in detecting star 18, star 10 alleles. Only PyPGX from the short read data was giving a hint of having some potential alternate phasing, potential novel alleles. And actually what we saw with long read was that it was not star 18, star 20, but star 16, star 18, and an additional SNP. So we can see that over here again. So for first haplotype, that is star 6, this corresponds to this SNP over here and this SNP over here. And then for haplotype 2, actually, it maps to this star 18 SNP down here, which you can see here. But also there's this additional SNP that's phased with it that's not attributed to any, it doesn't align actually with any of the known star allele definitions. It overlaps with the star 20 star allele. That's why it was appearing over here. But actually the phasing does not correspond to star 20. And we can see the SNP over here, nicely phased with the downstream SNP over here. So again, another example in this case where phasing actually led to a potential novel allele. Okay. Now, I've mentioned that we came across 2 discordant calls, so just to share about those as well. The first one was a multi-allelic site in ABCD1. So for this sample, we were using short read data as the truth set. And you can see over here how short read data was reporting star 1, star 2. So star 1 is defined by the presence of this C allele, which is nicely supported by the short read data. Star 2 actually is the A allele, not quite aligned with what we see. Actually, again, this is a multi-allelic site, right? So it's alter or heterozygous for the 2 alternate alleles. So after manually reviewing this, it shouldn't be star 2. It's a different allele. But regardless, from the long read data from our long read assay, we were still returning an incorrect call for star 2, because Claire 3 in this case was returning a homozygous alt for the T allele. It's not, when we looked at the raw data, we could actually see some signal for the C allele, so in a way quite aligned with the short reads, but the depth at this point wasn't enough to call this as a head variant. So this is something to refine in terms of, again, going for a bit of higher depth. And then another similar example was for F2. So over here, GetRM, microarray data, short read sequencing data are very consistent in returning star 1, star 3. And in our case, we were returning star 3, so hom alt for this T allele. When we looked at the PyLab of reads, actually, yeah, it was consistent with the T allele. But then looking at the phasing data, what we could see was actually that we were missing reads supporting haplotype 1. So again, related to poor coverage in this region. So that's actually something that can be mitigated at the level of running the test by setting actually some thresholds around the depth to interpret these locus. Okay, so I've shown results of diplotype concordance. I've shown some of the improvements, some of the discordant calls, but overall, how does long read adaptive sampling compare to other methods, right? So this is what we looked at here. We looked at a concordance of the diplotype calls as well as callability. And we compare results of the Illumina GSA chip. We compare results of short read sequencing data analyzed with 3 different methods, and then results from our long read workflow. And what we could see in a nutshell, just to summarize, is that for the long read workflow actually was outperforming the other platforms in 13 out of 20 genes. These are the ones highlighted in blue. But overall, we could also see that for microarrays, and here you've got highlighted in bold the cases where the metric drops below 95%. So microarrays was really struggling the most to resolve diplotypes in all of these genes. And fair enough, this is just a rather general microarray chip. There's other more specialized chips, but this is the data we had available for this type of analysis at this point in time. So microarrays perhaps those struggling the most. Interestingly, for short read sequencing, what we saw was quite a bit of variability in performance across the different pipelines, perhaps with Dragon coming up as a top performer. However, the one thing we noticed with Dragon as well is that in some cases it was able to reach 100% concordance at the expense of returning no calls. So at the expense of lowering callability, which in a clinical setting, actually, it may be the preferred thing to do. But on the other hand, as I mentioned, for adaptive sampling, we saw that there isn't so much of this compromise. We were able to get overall good concordance without much penalization in callability. Okay, and then the last slide in terms of the accuracy study, we also looked not just at the diplotypes, but at the phenotype concordance. And this is quite important because this is how results will eventually be used. They will be interpreted into a metabolizer profile, and that's what will be used to guide any changes in medication or in dosing. And so we've got the same 3 sample sets as before. And in a nutshell, what we can see is that overall, we saw very good concordance with the expected phenotypes. And we particularly looked at those calls that had been previously flagged as improved diplotypes, because we wanted to see for them what happened with the phenotype. Did it remain concordant, or perhaps it remained something that couldn't be interpreted anymore if we're talking about novel alleles? So this is what we saw, actually. We saw that the majority remained concordant. So even though there was a refinement of the diplotype call, it didn't lead to a change in the phenotype. But actually, we saw a significant number that after refining the diplotype became indeterminate. And this can be a bit of a double-edged sword in terms of having a more accurate assay. That you can be more accurate in terms of diplotype calling, but then if you identify a lot of novel variants for which the function is unknown, it also limits the clinical applications or the next steps of these variants. So that's a potential follow-up point for future research to do a bit more characterization of these novel alleles. And then we also saw, interestingly, some improvements in the phenotype calls as well. So not just the diplotype, but also some refinement of phenotypes in the right direction. So just to give you a sense of what that looked like, this is actually a sample from our clinical partner lab, TTSH hospital here in Singapore. And this is an example for CYP2D6, where they were able to identify using microarrays these 2 star alleles. They were able to see that there was a copy number change, but they couldn't resolve it. And as a result, the phenotype interpretation was indeterminate. However, with long reads, it was possible to actually see that it was a star 2 allele that was duplicated, and so then that gave an ultrametabolizer phenotypic interpretation. So this is what I meant by in terms of leading to improved interpretation. Okay, so now entering the last part of my talk, I also wanted to share the results of the precision and specificity studies, even though they may be less exciting, still much needed to establish a clinical assay. So very briefly, what we saw in terms of precision, so we looked at a couple of things. We looked at reproducibility, just looking genome in a bottle samples. We looked at replicates, both within the same run and across runs. And overall, we saw low variability as measured by coefficient of variation, and similar story for repeatability. When running the same analysis from the same inputs multiple times, we actually saw 100% consistent results. So high reproducibility, high repeatability of the assay. For specificity, we did also a couple of things. So we were interested to see whether there was any possibility of interfering substances. In this case, because those were blood samples, we decided to spike in triglycerides to try to simulate someone with very high triglyceride level And yeah, these, just to emphasize, we simulated very, very high triglyceride levels. And interestingly, what we saw was that actually for the samples that had the spike in, the DNA concentration after extraction was heavily reduced, almost by half. And that it was reduced to the point that it wasn't possible to continue with the sequencing. So that is a bit of a learning that for this type of samples, there may be an increase in the failure rates of the assay. Again, something to follow up on, perhaps with other interfering substances and with other levels of triglycerides that are a bit more close to what one may also find in the clinic. When it came to cross-contamination, we ran this external software, some of you may be familiar with, verified by ID, and we consistently detected no contamination across the study samples. So again, all of these supporting the idea that this workflow could be implemented in a clinical setting. Okay, so that was the majority of the talk focusing on the on-target signal. I wanted to wrap up by also going back to the off-target signal. If you remember, those were the small fragments spread genome-wide that we were also getting just because we need to identify for every DNA fragment that lands in the pore whether it's of interest or not. So what we did over here, because it was averaging at around 3x, was we look at one specific genome in a bottle sample, and we did imputation using this off-target signal against 1,000 genomes data set. And what we were able to see, I've got the results here in this plot. So I'm showing the imputation accuracy on the y-axis, stratified by allele frequency. We have up here in light blue the results of the imputation using the off-target signal. For this specific sample, it came out to be at 2x, not 3x. But what we were able to see was that this imputation accuracy actually was quite high throughout the allele frequency range, especially when compared to microarrays. We could see consistently higher accuracy, which makes sense because for microarrays, there would typically be a selection for more common variants. But then we were also able to see that the performance of the off-target signal from long read sequencing actually was on par with what you would be able to obtain with short read sequencing data at about the same depth. Both of them outperforming microarrays. Okay. So with this, I'll just wrap up. Hopefully today I've been able to show some of the work we've been doing with long read sequencing, how we're using specifically adaptive sampling from ONT for PGX testing, how we're able to achieve on-target depth of around 25x, off-target depth of around 3x, with relatively low DNA input. So that is some tweaks, some modifications we did in our protocol. And not only that, how this type of data allows us to accurately call both small variants and large variants, but also accurately resolve diplotypes and phenotypes for key pharmacogenes, in some cases with superior performance compared to other methods. And also, hopefully, I've been able to show that adaptive sampling, it's perhaps unlike other targeted methods, still retaining quite a bit of genome-wide signal that can be used together or by using imputation that can be used for downstream analysis. So all in all, yeah, I've been talking about, we think what I've been talking about represents for the first time a potential to clinically implement pharmacogenomics using long-read sequencing. And I just wanted to close by thanking the team, specifically the team at Nalagenetics, Pam, who's been leading the bioinformatics analysis, Han-Lin for the wet lab work, our collaborator, Dr. Ling at TTSH, for the help with the clinical study, providing the clinical samples, our collaborator at NUHS, Prof. Elaine, for the discussions, our funders, and of course, yeah, everyone here for their time. Happy to take any questions.
Thank you, Dr. Ma, for the insightful sharing about the pharmacogenomics testing with long-read sequencing. Now we will move into our Q&A session. You can unmute yourself to ask a question or feel free to type your question in the chat box. Does pharmacogenomics predict cancer and recommend the most effective drug for each cancer patient?
Okay, thank you so much for the question. All right, so I'll take it in two parts. Does pharmacogenomics predict cancer? Actually, that is not the primary goal of pharmacogenomics. Pharmacogenomics is about response to treatment, response to drugs. But it is possible to develop risk prediction tests based on the method I've presented, adaptive sampling. Actually, this is part of our ongoing research work as well, where there may be different biomarkers that may lead to higher susceptibility for cancer. I think some of the well-known ones are highly penetrant mutations, for example, in BRCA1, BRCA2, that may predispose you to breast cancer, but also polygenic risk scores, right, where you have multiple contributions or small contributions across several hundreds of markers, if not millions of markers across the genome. So actually all of that you can measure also with the adaptive sampling method I've spoken about. It would be a different application to PGX. However, that said, PGX can actually help to recommend effective drug treatments for cancer patients as well. So we've done a few studies around that. One of the recent ones that we've been able to publish with some clinical collaborators in Indonesia is actually looking at tamoxifen therapy for breast cancer patients and how it is possible to adjust the dose of tamoxifen depending on the profile of mutations in CYP2D6. So yes, absolutely, pharmacogenomics can guide prescription of cancer drugs.
Thank you, Dr. Ma. We have another question. How about the cost comparison on short read sequencing and long read sequencing?
Yes, yeah. So thank you so much for the question. Yeah, there's it's a tough question to answer because of course a lot of that depends on the volume. So I will not be able to provide exact numbers. They will be very much lab dependent, but actually we are trying to push it. So I'll explain what we're doing at NALA is we're trying to push the cost of the adaptive sampling test to be as cost effective as what we're offering right now with microarrays. So we're not there yet. Right now, we are running adaptive sampling using 3-plex. In order to get there, we have to push it a bit further. We have to get to a 6-plex or beyond, but this is our target. So it can be, I'll say, it can be quite cost competitive, but of course, it also depends on the volumes, right? If you're a facility running millions of short-read genomes, then yeah, your price point, your starting price point may be quite different. Conversely, if you're a facility running millions of long-read samples, you may also have a different starting price point. So yeah, unfortunately not an easy answer, but I'll just say it can be quite cost competitive to use adaptive sampling.
Okay, thank you, Dr. Ma. Yeah, Dr. Ma, we have another question. In case of mixed races country, will reference genome for pharmacogenomics be problematic later?
All right. Really good question. So there's different ways to go about it. So one of the things that you may see is a different distribution, different frequencies for the known pharmacogenes. And this is something that population sequencing studies, for example, are well in a good position to address, including the PRECISE program here in Singapore. They've been looking at that, where you can see differences in the most prevalent star alleles depending on the population. So this is one. So that's quite relevant if you're running a targeted assay, because then you want to make sure that you capture those more prevalent star alleles. But then this is based on the known variants. There's also the unknown, right? And as I pointed out, the majority of PGX variants are rare. And there's a lot of potential to uncover novel variants, as it's the case with a lot of the genomic variation databases. They're heavily skewed towards European American samples. So yeah, there's a lot of potential to uncover new variants. We did a bit of a study with looking at CYP2D6 variation in a Singapore cohort of 10,000 samples, and we saw about 2% of novel variants there. So you can see how that quickly scales. So in a nutshell, yes, it is an extra consideration to take into account the local ancestry. Yeah.
Thank you, Dr. Ma. Another question is the error rate for nanopore higher compared to NGS or microarray for pharmacogenomics?
Oh, right. Okay. Okay. So there's different things that you can look at. You can look at raw quality scores. I think that's a comparison that people often do, right? Between we hear about base quality between short read sequencing, long read sequencing, and perhaps long read sequencing methods being labeled as not being on par. I would say there's been a lot of improvements on the long read sequencing space, both in terms of chemistry and algorithms, like the two base calling modes I was mentioning that really boost the raw rate or the underlying base calling performance. But then that's not the end of the story. That's just an input to the whole workflow. And what we've shown here, actually, hopefully with this work, is that at the end, when you look at the variant calls, when you look at the diplotype and then phenotypes, actually there is not just that long reads to work, actually that they can improve upon other methods because of all the other advantages. So having access to longer reads, which may result in better phasing and so on. So yes, so and then perhaps the other thing that I'll mention is with regard to the off-target signal. It was also quite telling to see that even at 2x, of course, combined with imputation, but there was virtually no difference between the long read data that potentially had a lower quality and the short read data with a higher base quality. So yeah, so these differences actually become mitigated as you start looking into downstream analysis.
Thank you, Dr. Ma. Is there any more question from the audience? I believe there is no more question. So thank you so much to Dr. Ma for the informative and engaging presentation today. And also a big thank you to the audience for attending this session. We hope you find the information is valuable and inspire you to apply it on your own research. Before we end the webinar today, we would love to hear your thoughts. Please take a moment to fill out the survey form in the chat box. Also, if you have any question after this webinar, please feel free to reach out to us at sales@nextgene.my, or you may contact our lovely product specialist. Thank you once again, and we will connect. And we are looking forward to connecting you in our next webinar session.